Hello everyone, I'm Mingbo Cheng, currently a PhD student from the Institute for Computational Genomics. Today I bring you PHLOWER, a powerful trajectory inference method using graph Hodge Laplacian decomposition. Nowadays, many methods have been proposed to account for trajectory inference. However, for complicated data sets, many methods failed to capture the dynamic process accurately. On top of that, almost all the methods only consider the signal on the vertices of a graph. For example, for diffusion map, it decomposes the transition matrix of graph Laplacian and uses the spectrum to reorder the cells. But we can use some other information, for example, the signal on the edges of a graph to get more accurate trajectories. With this, we have the high order graph Laplacian, that is graph Hodge Laplacian. They are also high order of instance matrix like B1, B2, B3, etc. Then we have also L1, L2, L3. With this, we propose PHLOWER, which take a single cell count matrix as input and output the trajectory trees using stream to visualize. To demonstrate how Hodge Laplacian works, assume there are 3 islands in the sea. If a whale wants to chase these fishes, it can take the paths like 1, 2, 3, 4. Can we build a model on this? So these trees, this island can take as obstacles, so we can place holes in here, and then we can create a network. So then we can decompose L1 to get the information of this network. So we use a simple data to show how the Hodge decomposition looks like. Here, this data that we have 2 holes. As we can see, the first and the second eigenvector selects these 2 holes, and interestingly, the corresponding eigenvalues are equals to 0, and these eigenvectors are called harmonic functions or harmonic. If we project trajectories onto this harmonic space, as we can see, the trajectories at different sides of this graph, like these orange ones are on top of these 2 holes, the green ones are in between of these 2 holes. They can clearly separated in this harmonic space. So we can perform trajectory clustering on this in harmonic space for these trajectories. Also, we can use cumulative trajectory embedding to capture the dynamic of each trajectory. This is a really simple data set. For single cell data, we have tens of thousands of features. We applied several tricks to create the graph with 2 holes for this single cell data. So then we can apply Hodge Laplacian decomposition. As we can see here, the first and the second eigenvectors, and they are harmonic functions because the eigenvalue is equals to 0. We also develop a preference random walk and project these trajectories onto harmonic space. As we can see, the trajectories can be clustered really well in this space. When we look at the cumulative trajectory embedding, we can find that at some certain point, trajectories goes to different directions, which indicates that we can use this cumulative trajectory embedding to find the branching point of the trajectories. So with this, we eventually create trees using the cumulative trajectory embedding. So this is the workflow of PHLOWER using a really simple single cell dataset. When we encounter a more complex dataset, for example, here we have a metal policy dataset. As we can see, we use cumulative trajectory embedding account for the branching point. Eventually, we create a really beautiful tree. Okay, that's all for the PHLOWER. So finally, I would like to thank my supervisor, Professor Ivan Costa, and all who contribute to this project. Thank you. If you have any questions, please feel free to contact me.